Okay, today we're going to present on induced pluripotent stem cells. Oh my God, there's a typo there. Anyway, so induced pluripotent stem cells is a kind of pluripotent stem cells, which is a kind of stem cell that can differentiate into almost any cell type in the body. Yeah, the most well-known stem cell is embryonic stem cells, but today we're talking about the other type, which is induced pluripotent stem cells.
Also, guys, feel free to interrupt and ask questions during any of these.
Okay, so the difference is that embryonic stem cells is directly derived from an embryo, but induced pluripotent stem cells are generated by reprogramming adult somatic cells to a pluripotent state, which basically means that you can just get a cell from any kind of cell and then reprogram it back to the pluripotent state, which can differentiate into different cell types. OK， 然后这个是它的 develop timeline， 然后在iPSC 这个东西，它是最先在2006年被一个叫 Yamanaka 的日本科学家给发现的。然后呢，然后后面就 Can I zoom in? OK, I can. 对，然后Anyway， 我们先下一个，就是一开始的这个stem cell 呢，它是一开始他们是在老鼠里面发现了，如果用4种 transcription factors， 就是 OCT34 SOX2 KFL4 and CYMC， 然后他们就可以把老鼠的一个 germline 去把它给reprogram 成stem cell， 然后呢后面他们把这同样的4种 transcription factors apply 到 human 的 fibroblast 里面，然后他们发现这个4种 transcription factors 也可以把 fibroblast 给 reprogram 成iPS cells， 然后呢，这个发现让他们在2012年获得了诺贝尔生理学和医学奖。其实它的 production 它的 theory 是比较简单的，其实就是你把任意一个 somatic cells 从你的身体里拿出来，一般大家会用 fibroblast， 因为它比较 sturdy， 因为它其实是一个肌肉细胞，然后呢再去把它给用这个所谓的 Yamanaka factors， 就是上面刚才那个 slide 说的4种 transcription factors， 去把它给就是 introduce 进去，然后呢这些 transcription factors 就会让这个 cell 去把自己给 reprogram 成一个 pluripotent state。对，然后呢 introduce 的方式一般用 viral vectors， 然后呢现在也在去发明一些不是 viral vectors 的，就是没有那么不 integrate viral 的一些方法。然后接下来就是 selection, expansion and validation to make sure that the cells that are reprogrammed back to the pluripotent state has the ability to self-renewal and then also differentiate into the target cells. 然后呢 application 其实有非常非常多 application， 我就是大概选取了4种。第一种就是 disease modeling， 其实 iPSC 它是 serve as a tool， 它自己就是并没有说有关于它有太多的 research， 当然也有很多，但是就是它 serve as a tool 会有更就是广阔的应用。第一个就是disease modeling，so you can use iPSCs to let it differentiate into different cell types and then to mimic different kind of diseases. And the most widely known are neurodegenerative diseases and cardiovascular diseases, and diabetes are also included. 然后第二种是drug discovery and testing， 基本上就是 personal medication， 然后这个现在没有说有特别多的应用，然后 clinical trials 也是感觉是比较停滞不前。然后 regenerative medicine and cell-based therapies 就是差不多。然后呢，第4个是 tissue engineering and organ engineering。然后现在是 organoids 这个 topic 非常的火。然后 organoids 是 miniature simplified version of organs like liver, kidney and brain. And these organ 就是你可以把它想象成一个非常微缩版的一些 organ。然后呢，在这个 organ 在这个 organoids，你就可以让科学家在like lab-based settings 里面去研究一些 disease，就不用去搞一个 like actual organ。然后呢，这个右边这个图是一个 brain organoids，然后呢，它就是把 like different cell types to integrate into a tissue and then build the tissue into an organ that functions similarly as the actual organ。然后呢，clinical trials，一开始，应该是在2012年的时候，然后有一个retinal 的organoid 就被 build 出来，然后后面在2017年的时候，他们就这个 clinical trial 也在日本，然后他们就把这个就是他应该是有一个 healthy donor，然后去 donate 了一个视网膜细胞，还是like similar something like that。然后呢，去把它给 reprogram back to 一个pluripotent stem cell，and then 然后就把它给就是手术移植进这个这个患者，他是叫黄斑退化，应该是它是 age-related macular degeneration。然后呢，我看资料上面说，就是其实这个 surgery 它并不能帮助他的视力去就是 come back to normal level，but it can help with like control further degeneration of existing cells。但是好像后面说也有 rejection 之类的 complications，所以好像也不是特别顺利。Okay, future direction. I feel like the most promising and popular one is immunotherapy and cancer treatment like CAR T, and also gene editing. 好像现在有，Wait, what happened? OK，现在就是gene editing 比较应用，比较就是正在做的一个research 是关于就是他们就会把Wait, what's that disease called? Anyway, I'll update you later. 对，然后第三个就是 tissue engineering and same thing serve as a tool and then etc. Okay, that's it.
我想补充2点 for 无意思的那个 slide，有一个是就是最近新出了一个新闻是，就是中国的一个女的是25岁的一个女生，然后她在以前是有 diabetes，然后 I think she received like she got like her own cells reprogrammed into beta cells and put it back into her body. And then it's been a year since she received this like cell transplant, and she's like, she no longer has diabetes now, and she can eat sugar and everything. It like just came out a few days ago. Very astonishing news. It's on Nature. And they just published their results on Cell, so it's very cool. Another thing is 先前武一讲到说，就是有4个那个 transcription factor，可以就是reprogram into like iPSC, reprogram any cells into iPSC. I just want to say that their group also did another research where it's like, 如果你只把这4个 transcription factor added for a set amount of times, like a very short times, it can actually rejuvenate like old cells into like that behave more like young cells. So a lot of people are doing like age reversion or like rejuvenation experiments with these transcription factors.
是外泌体吗？是外泌体吗？
什么东西？
就是你刚刚说的 rejuvenation 的那个。
对，就是它会让这些 cell 变更年轻一点，就是他们的，比如说它 transcription like profiles 这些 cells，但是 it's very interesting because it doesn't work。就我们的 lab 有在做类似的实验，然后我们 lab 是用HSC 就是 hematopoietic stem cell，然后加这4种 transcription factor，想想看会不会变年轻，但是好像就对 HSC 没有什么用，但是。
就是那个人一直没有投入他那个 trial，主要的原因是其中一个 factor， 它是一个 oncogene factor，
对
c-Myc 是它确定的会 high induce cancer 的一个 factor， 所以它就很难真的用于人身上。
就是说它
是3个正常的加上一个 oncogene factor。
对抗衰有用吗？
well， 就是可能 potentially trade off 是你是抗衰了，但是 you got the cancer, you got
It's a bit of a risk.
我 add on 那个 gene editing 那一 part，就是他们现在在用这个在用 iPSC 去处理 sickle cell anemia，它不是直接去治这个病，而是把就是edit 过的 iPSC 去放到小鼠还是身体里面，然后就让它们去产生正常的红细胞。 OK
OK 好 Yeah, so basically, 我今天来讲的这篇 paper 是 Kyle Low Lab at Stanford, very young PI, got his PhD with Irv Weissman, who is like the biggest person in the stem cell field at Stanford too. So basically what they did is they generated human artery and vein cells from iPSC, and then it kind of they use these cells to investigate what Nipah and Hendra virus do to endothelial cells, basically. Okay, yeah. So this is like a basic introduction. Endothelial cells is like crucial for vascular functions, and artery vein cells have are part of are like two distinct types of endothelial cells. And then one of the challenges in this field is that it's very hard to generate pure populations of artery and vein cells from human induced pluripotent stem cells. 就是先前的这些就是programming， 比如说加各种factor 进去 induce 成这个都会是不是 pure 的 artery 或者 vein endothelial cells， 他们会有很多其他 mixed endothelial cells, but that's not very helpful. So what they want to do in this paper is to generate pure population and to understand the signals that control these subtype specification. And they also are interested in studying Nipah and Hendra viruses. These are two lethal paramyxoviruses with very, very high fatality rate. And it's like prevalent in Asia and Australia, I think. And they cause severe like vascular diseases. These are also very, very dangerous. So you need BSL-4 containment. And then there's no approved treatments or vaccines for human use. And so it's usually very hard to study these like viruses because of, so first they're like lethal. So, and also they are very pathogenic. So, 就是比较难去就是 study 这些viruses. So what they want to do is they want to differentiate these in vitro artery and vein endothelial cells so they can study these viruses with these in vitro derived cells in the lab, right? Yeah. And this is just a picture of the BSL-4 labs. I thought it's very interesting because I've never seen a BSL-4 lab. So yeah, 就是他们要穿这个才能进这个 lab， 非常有意思。But yeah, so this is kind of like the schematic of how exactly they turn these induced pluripotent stem cells into artery and vein cells. So for artery cells, they did like a 就是他们先把它 derived 成了 primitive streak cells. So these are part of the fetus developmental program. 就是你会先变成 primitive streak, 然后再变成 lateral mesoderm cells. So these are all part of the normal fetal development, right? 然后变成 mesoderm cell 之后，然后他们再用一定的这些 transcription factors 去把这些变成 artery cells. And they also tested a lot of different combination of things basically. So basically, you need certain signals that promote the induction of mesoderm cell to artery cell. You also need certain signals to inhibit these mesoderm cells to differentiate into other cells. So it's you need to try different combination of these chemical cocktails in order to make it work. But yeah, and they very surprisingly generate very pure populations, like 70 to 80% just all artery cells. And kind of like similarly for vein cells too, they found this like signaling pathway called ERK pathway that it needs to be temporarily activated and later inhibited in order to produce these pure population of vein cells. You can see here that so these NR2F2 is a vein marker, and you see that the purple one is has the highest vein marker, and the purple one is if you only inhibit ERK on the 4th day of differentiation, and you let it be from D0 to D3. And it's very interesting because in previously this has not been described in any vein differentiation protocols. So it's just like they are simultaneously discovering new important developmental signaling pathway while differentiating into these vein cells or cells that they want. And this is very cool because this iPSC-derived artery and vein cells actually demonstrate functional integration and vascular network formation in vivo. So I think when reading these iPSC paper, what I always look for is if they have in vivo experiment, like these cells like work or like seems to work perfectly in vitro, but the challenge is always like 你把它变到 like in vivo system 里边的时候，它能否 adapt to the in vivo system, right? And that's like what matters because that's eventually you want these cells to have therapeutic purposes. And I think the in vivo part is like crucial to that therapeutic translation in the future. And very nicely, they found like in vitro, they found like nice transcription profiles that match actual artery and vein cells. And then in this CND, 他们把这个 transplant 到了就是immunodeficient 的 mice 里边，然后这些也 form a very nice 的这个 artery network and vein network in 3D environment and in vivo system. And they're all like functional and doing great jobs. So I thought that's very interesting and important too. And so thirdly, they study like how Nipah and Hendra viruses kind of like interact with these cells, right? So 刚刚我一讲了说有一个 important significance of iPSC is that 你把它们 differentiate 到某个 cells 之后，它们可以作为就是in vitro 就是 study 某一个 disease 的一个 models, right? So they are using these in vitro cells to look at Nipah and Hendra viruses because it's very poorly understood. So one thing that they noticed is, interestingly, Nipah and Hendra viruses all target artery cells more than vein cells, and they form these like syncytia, which are cells with a lot of nuclei. 就是这些 cells 都 fuse 到一起了，所以一个 cell 里面会有很多 nucleus， 然后到最后就直接就 burst， 然后 cause cell death. So that's basically how they target the arteries in when they 就是入侵这个他们的 host. And then another very interesting thing is that so we have some people in immunology class here. Something very interesting is that these virus usually 就是正常的这个 virus 入侵到一个人体内，它会 induce interferon responses, which is like innate antiviral responses that the host have. 这是我们自己 protect 自己的一个 program, right? 然后我们可以看到，首先one thing that says 就是 Nipah 和 Hendra virus 它们可以就是他们会入侵到 host genomes， 然后so like it make the cells， 就是他们可以快速的让这个 cell 的所有的 genome 都incorporate 到这个 cell 的这个 host cell 的 genomes， 然后让 host cell 都replicate 他们这个 virus 的 genome， 这是他们的一个 strategy. And very interestingly, they induce no interferon responses compared to Sendai virus, which is non-pathogenic. 也就是说，他们可以 evade 这个 immune response， 自己的 antiviral response 就非常的厉害。然后 in contrast to Sendai virus, which is a non-pathogenic virus that can activate robust immune activation in the host， 然后 host 就有自己的这个 program 去 defend 自己。然后这两个 lethal virus， 他们就是你的 host 很难去 defend 自己，因为他们会逃脱这些自己 host 的这些immune activation and stuff. So it's very interesting. And this is, of course, not known before. Yeah. So yeah, basically they develop this protocol that generate pure population, and they prove this in vivo. That says they are very like functional, and they also say something about these viruses effects on these two cells. So yeah, that's basically it. 然后 Vicky 会给我们一个 industry 的一个
overview thing.
我把它喂，stop share。stop。应该在，我鼠标在哪里？哦，回来了。OK，我 stop share，让你来 share，
让我来 share， 然后。
那
你这个把给
因为我不知道我把没有试过我这个电脑投屏，所以不知道该怎么办。
所以要
我可能让你来。
Oh, yeah. Okay.
我可以整体上的你看这样有封面吗？有的，
就是 double click，你就单纯 double click。
No，我看我
把它那个
high，我想 high speak。哦，
再拉。
是这样。
Yeah，就是一个 generally overview. And I have to appreciate my professor. 就是我们刚上一节课，所以 actually most of the slides is from the lecture notes. Yeah，但我觉得他是做的很好，很清晰，so it's good for share. So there are currently all of them are currently what FDA approved as cell therapy and gene therapies. And most of them are in hematology. And then the second round would be oncology and then dermatology. And then like they're less immune and less for the muscle stuff. So that's like an overview of stuff. And then
Are these just cell therapies or specifically stems?
they're more like combined cell and gene therapy. Now, like cell therapy usually use iPSC as a function, but it's not guaranteed all of them using it. So this is general how people like if it approved what people do to gene and cell therapy. One thing is they do ex vivo, so they collect what from donor or from patients, and then they program it and then reculture them, and then they deliver those engineered cells into the patient. Another in vivo way is more like you're using some virus like AAV or LNP, and then you can either local injection, like injection to place that have the disease, or you have symptomatic injection. It's more like probably through IV infusion, and it go to all of your body to cure this disease. So and as the bottom listed here, they have the approved ex vivo drug and the approved in vivo drug. So this is more like a comparison between how ex vivo works and in vivo works. And this is also like from FDA, how currently there are 340 biological medicines that are approved, but only 32 are cell and gene therapeutics. And we can tell most of them are cord blood driven. And the second would be like cell therapy. The one that harder are gene therapy and CAR T therapy. The reason is because they require a high technology background of the company, and also because they need more money. Way more money than cord blood driven therapy or other cell therapy.
想问一下，那个 cord blood derived 是自己的 cord blood 还是别人？
是 donor.
OK，
对，都会。And then there are some approved cord blood therapies. There's like a lot. And then there's also what I approved for CAR T therapies. So CAR T therapies is currently like a increasing field for lots of disease, especially for cancer, because lots of patients are in response to chemo or to immunotherapy. So CAR T therapy is a way that using their own cells and then design their own medication for those patients. But cord blood therapy has a problem is because cord blood is so little, so it usually can't cure adults. So mainly it can be used to cure pets like 3 or 4 years old. They can use this way to get cured. But if you're like an adult, like you're 18 or 20, then probably cord blood therapy is not the case. But also some people like in China or some other places, they ask you to storage your cord blood when a newborn baby has it. But the thing is like it can only prevent the baby for any disease like before 10, but after 10, it is not worth it anymore. The reason is because the dose is too little. So there are a list of how FDA currently approves cell therapy, and we can tell like for autoimmunology, like auto means your own cells and then inject it back to yourself, and then allelogenic is more like you got the cells from a donor and then inject to your body. So they're like half and half. So 5 out of 9 are auto and 4 out of 9 are LL. And then you can see the description on how they're going to use them. And lots of them are using as a scaffold. So there can be either like in a surgical way. For example, if you're on a surgery and then the physician cut off your organ for some parts, and then they can put a scaffold that contains those cells and helps the healing, or it can help like local healing or trying to kill this disease. And also another scaffold thing is like lots of people get burned, so their skin cannot regenerate by themselves. So lots of them are using the scaffold as a way to make the skin repair.
想问一下，如果是比如说你说cellularized scaffold for like during surgery，那这些 cells 是 fully differentiated 的，还是 progenitor，还是更往前
？Usually 他们会用 iPSC， 然后让他们大部分变成 stem cell， 但是他们有一部分是 fully differentiated， 然后他们会有一个就是那种 physical 的东西，让他们往他们想要的方向去 differentiate。就是他们有些会有涉及到那种 materials 的东西，或者是那种 physical structure，这样他们就会去找。然后很出名的一个东西就是说，你的膝盖，因为哥大有很多就是做膝盖治疗的，就是如果你的膝盖或者老了以后它有磨损，所以它会空了那块就没有胶原蛋白了，他们就会做一个手术，然后有一块 scaffold， 那个 scaffold 有些就会有一点点 cell。就是一点点 stem cell， 它可以是你自己的，但是因为在这 scaffold 里面，它会让这个，由于它 scaffold 的设计，让这个 cell 去更好在这里面生长，所以当它被安装在你身上了以后，它只需要一点点就可以把整个 scaffold 都长满，然后你原来那一块洞就填不上去。对，然后 usually scaffold 它会是 biodegenerative， 就是随着时间的推移，它就慢慢被你消化了。
所以 scaffold 是 provides a like microenvironment for the cells to grow。
然
后放进去的还是 stem cell。
放进去会放一点点就够了。
OK。
因为有的时候它特别是有一些就是类似于你有 time concern 的东西，它没有办法一下子生产足够的 dose， 所以它会用一些其他方式。
因为我们就是老师经常说，就是说他们一般 inject 就不敢 inject，就是有 high potency 的 cells， 因为it's very prone to become cancer， right？
然后这是一些就是 CAR T therapies 目前但它就是少很多。然后 usually 就是看就是 CD19 这是一个比较 well known 的 target， 然后大部分其实 CAR T 用的是 B cells， 就是因为 B cells 属于是 immune system 里头它可以干活的，可以记忆，就是比较好用的 cell。这些的 ，and those are some companies。And then, but also bad news are， 比如说今年4月份的时候 ，Sanofi 就 close 了一个就是 我简称它为 iPSC， 就是这个它是专门做细胞治疗的。然后当时赛诺菲把它收购的时候花了3亿美元，但是就是还是由于造价太高，它没有办法负担剩下的经营，所以就是这个收购的公司就被关了。然后同时的话，5月份的时候又关了一家，就是 Engineered Cell Therapy， 原因就是因为他们在 B 轮融资的时候突然有一个说要撤资，所以他们接下来没有足够的资金再走下去，所以也就关掉了。对，所以就会有，其实现在有很多那种初创公司很好，但是他们不一定能走到 FDA approved 的阶段，那就会由于各种原因就关了。OK，and that's all what I think。投资有风险。
大家有什么问题可以
讨论。
Open discussion。那我觉得你说。
有一个 slides 可以分享给大家，这是一个补充，就是 how iPSC 可以被driven到不同的。
刚刚提到那些 FDA approved drugs， 他们的一般的造价是多少？
造价康明没有说，但是你可以有销售价。
那销售价一般？
销售价一般在100万。like $1 million美金。
Right, yeah.
但是它的有一个问题是它 一些 one dose 的就会更贵，one dose 可能是500万到700万期间，for one dose 就是你得一针然后你就好了。但是像100万，就是它可能是 multiple time，所以它如果你比如说治2年，那也就是200万。
哦
对，而且它就是而且有的是说法是，因为你一针 one dose 可能就是 like 10几万，但是你一年可能要 multiple dose。So it's like crazy expensive。
造价也许由于它的那个算不算研发费用？研发费用
研发费算的很贵，
就是如果不算研发费用的话，大概是20倍以上，就是它的卖价的20倍以上。如果算研发费用的话，过百倍很正常。
哦，对，我觉得之前很好玩的一个 science 让我分享一下，就没注意在踢球叫什么？
哎，能那个去有一个 slide 能发群里吗？就是说那2个公司破产的事情。
哦，呀，
先入为主，看一下这个领域就破产很家常便饭，但是看看是怎么定义破产。
OK，就是这个就是其实是 add on 就是5一的一个 background，就是我们上课有讲它 polypotent 可以变成 multipotent，然后 multipotent 就会变成你 differentiated cells，但是现在有很多人在最快的方法就是上课有讲到一个，它从 fibroblast 直接变成了 neuron，所以它其实跳过了还原变成 polypotent，再变成 multipotent 的这么一个过程。然后也有很多变成 multipotent 的话，就是它不一定要变成 polypotent，所以有很多，比如说这条绿线，就是它从 fibroblast 变成了一个 polypotent，然后再从一个 polypotent 变成了一个 neuron，就是 like it all can works，就是你可以 play around with it，对，但是其实效果最好的还是 polypotent，因为它有一个原因，就是因为大家会有的 research 认为 cells 有记忆，所以就是你去往 polypotent 里，它会更年轻，然后效果会更好，就是 focal 来说。但是你往 multipotent 其实可能疗效不如 polypotent 好，但是它其实少了很多的成本和时间。
等一下，multipotent 指的是它已经 differentiate 到那胚胎3个层的那个程度，还是说 it's even later？
So pluripotent means that you can it can differentiate into like any cells except the placenta,
Placenta.
right? Multipotent 就是指 pluripotent 之下所有的构造 between differentiated cells and pluripotent.
OK，
So 只要可以 differentiate into more than one cell type.
这是 immunology 吗？
For developmental.
哦，OK。
I click.
我本来今天要上的，然后
It's so fun.
这是我
It's not fun, but
course conflict。